Entomology is an important field of biology. Studying insects, their relationship to us humans, the environment, and other organisms is of high significance. Through this video, our group can share our experiences and newfound knowledge with familiarizing external body parts of insects, as well as their functions, collection tools, and various collection methods, and proper identification using identified morphological features. To familiarize the external body parts of insects, each member labeled basic identifying parts of insects and gave the importance and function of each part. This activity aided us in becoming more adept in identifying these parts in person with our collected samples.
In the laboratory too, we learned that it is important to make an insect bait that can attract more than one species to ensure the diversity during the specimen collection. In this exercise, we used the tuna as a protein source and peanut butter as a sugar source.
For proper active collection of insects, we use the following materials. First is Ziploc for the temporary storage of collected specimen. Second is the insect net, which we used for capturing the insect. Last but not the least is the kill jar that quickly euthanized the captured specimen.
The insect collection took place for approximately an hour in the garden area near the FC building at Far Eastern University, which provided a variety of habitats ideal for insect sampling. We employed both active and passive sampling techniques to gather a range of insect species.
For the active collection, the group spent around an hour manually catching insects using a sweep net and hand collection. The sweep net was particularly useful for capturing flying insects and those found on plants and bushes around the garden. The group also searched beneath rocks and within leaf litter to find soil-dwelling arthropods. For the passive collection, baited traps were set up for an hour to attract ants and other crawling insects, using ingredients available at home as bait. These ingredients included peanut butter, which acted as a sugar source, corn tuna as a protein source, mango seeds to attract granivorous arthropods, and dead spiders to attract carnivorous arthropods.
The general area where we collected is full of ornamental plants. A small balleted tree covers a good portion of the study area. Another interesting note is that the area is above a drainage hole, thus small green flies can be seen throughout the area. Collection, as mentioned, was done for roughly 1 hour and 14 different specimens were obtained, in which included flies, caterpillars, spiders, and dragonflies. After collection, the specimens were given repetitive IDs. This includes specimen 1 is a Plexipus species or jumping spider. Specimen 2 is from the species Artiinae or tiger moths. Specimen 3 is also an Artiinae species. Specimen 4 is a Sarcophaga carnaria or flesh fly. Specimen 5 is Musca domestica or domestic housefly. Specimen 6 again is another Artiinae species. Specimen 7 is Colaptera species or beetle. Specimen 8 and 9 are Tetranatidae species or long-jawed or beavers. Specimen 10 is a Psychotidae species or drainfly. Specimen 11 is another Artiinae species. Specimen 12 is a Solenopsis conformis invicta or imported fire ant. Specimen 3 is either Olepa or Aria species, which belongs under Artiinae. And specimen 4 is Pantala conformis flavescens or a wandering glider. Baiting, on the other hand, yielded different species of ants in only 3 out of 8 baits. Rotated identification was not conducted for baited ants.
Laboratory exercise number 4 covers essential entomological tools used in field collecting. Entomological pins are vital for mounting insects while protecting their delicate structures. A pinning block helps set specimens at the right height for proper display and storage. The puncher and the paper tips are used to secure smaller insects on the paper stage, which is then placed on the pin for easier handling. Water-based glue is necessary for attaching fragile parts like wings and legs without causing harm. Lastly, the Iguchi ball provides support during cleaning, preventing damage to the specimen. These tools are crucial for ensuring accuracy and care in insect collection and study.
So for the entomological probe, this is used for setting or dissecting the collected specimen, which is attached to the Iguchi ball. Iguchi ball setup. This consists of a cut ping pong ball filled with cement and clay. It is also supported by a bottle cap for stability. It also includes the pin specimen on an entomological pin along with its label. For the label or the specimen label, these include the date of collection, the area where the specimen was collected, and the name of the person who collected it. The order, family, and genus of the specimen are also noted. And lastly, for the insect specimen, this refers to the collected specimen that is pinned on an entomological pin and examined under a microscope to identify the type of insect.